2023 to give you a team presentation. So I'm Gisela, and together with my teammates, Jim, Cyrus, and Duncan, we're very excited today to present to you our project ideas. So let's first begin by introducing a bit of our team. So our team this year will be, so the project we're working on this year will be called KARMA. And basically what we're doing is that we're trying to deliver transgene into hard-to-transfect tumor-associated macrophage using self-replicating RNA as our factor, so in short, siRNA. So here is a quick preview of our presentation today. We'll be mainly talk a bit about the inspirations our project draws from, as well as a bit of the design and the data collection methods of our project. And in the end, we'll also talk a bit about the education and community outreach work that we have done in the past few months. So to begin with, it's very apparent that cancer is a very prominent health issue, even on a worldwide scale. In fact, it has cost 19.3 million. There is a 19.3 million new cancer diagnoses in just 2020 alone, and causes over 10 million deaths. And even if we scale down even if we narrow down our scope a bit and focus on Hong Kong only, cancer still presents as a major health issue. So there is over 30,000 new cancer diagnoses and over 14,000 deaths each year in Hong Kong caused by cancer, and among which liver cancer presents as one of the major culprits. In fact, liver cancer is the third most common cause of cancer-related mortality in Hong Kong and the fourth most common cause globally. And therefore, this is why it has become liver cancer is now an area that requires therapeutic breakthroughs, and also the reason why we chose it as the focus of our project. And since 80% of liver cancer cases worldwide were caused by hepatocellular carcinoma, in short HCC, this is also the reason why our experiment will be mainly using the cell line HT2 as our focus. This cell line is isolated from HCC patients. So now I'll pass the time to Jim to give you more details of our project.
Okay, so sorry, owing to rush of time, our platform might be a bit simplified, but I'll introduce you guys to our concept first. So we can tell that there are like different therapy for the cancer right now. So there are chemotherapy and radiotherapy, but owing to the intrinsic limitations, they are all having quite a lot of side effects for cancer patient. So instead, we are going to focus on immunotherapy. Yeah. So for the immunotherapy, there's quite a popular one called CAR-T right now, in which chimeric antigen receptor T-cell therapy. So basically what it does is just recognize tumor through specific CAR antigen binding, as you can see from the picture right there. And also it's a preparative agent that kills tumor cells. Okay, so I will now focus to the CAR first. So CAR is just a genetically engineered receptors that combines specific parts of tumor targeting antibody, the SCFV, with the T-cell signaling parts. Yeah, but for the mainstream CAR-T therapy, it still has some problem. So first, from the right, you can see that it can't traffic to the cell and penetrate the cell, penetrate tumor cell, because there is a tumor microenvironment surrounding it. So there might be some difficulties for the CAR-T cell to penetrate into the tumor and then bind with them. And also, the nutrient-restricted tumor microenvironment will lead to immunosuppression. So there will also be some M2 state in which pro-tumor macrophage that prevents the good macrophage, the M1 state macrophage, to act on the tumor cells. So it's quite difficulties for the CAR-T cells to pass through the microenvironment and then express their own function efficiently. And also there will be tumor heterogeneity. So for CAR-T cell, the basis of its work is to target the antigens expressed on the cell surface of the tumor, but not every tumor expresses the same antigen. So there might be cancer cells that express antigen A and antigen B. So for those who target antigen A, the CAR-T cell, they won't be able to target those with antigen B. So there is also a problem. Okay, so to tackle the aforementioned problems, we designed a project in which the CARMA, and in short, it will just be a self-polarizing CAR-M enhanced by RNP SIRPa knockout. Yeah. So I'll talk about the CAR delivered by siRNA first. So just Gisela just said, the siRNA is a self-replicating RNA, and we will talk about detail of it later. So the CAR we opt for it will just recognize the GPC3, in which a antigen widely presented on the hepatocellular carcinoma, in which the cell line that we use, the HKU, we also have this antigen expressed on the surface. And it will enhance the macrophage activation and phagocytosis. And also, by self-polarizing, the CAR-M cell we use will express interferon gamma, which will polarize the CAR-M into M1 state. So back then I just said that there will be M2 state and the M1 state macrophage. So the M2 state will be pro-tumor, which will suppress the act of the M1 state macrophage, in which the good one to act on the cancer cells. So by expressing interferon gamma along with the CAR delivery, we are able to ensure that the CAR-M is in M1 state, which can express its function properly. And also there will be a supportive measure in which the SIRPa knockout using the RNP. And we'll talk about it later as well. Okay, so the reason why we use the chimeric antigen receptor macrophage is because it can infiltrate the tumor microenvironment, which is something that CAR-T cannot do. And also, the macrophage can activate T cells as antigen presenting cells. So what CAR-T cell cannot do, CAR-M cell can do. And CAR-M cell can even notify the T cells and then cooperate as a whole in the system. Yeah, so it will be a schematic presentation of our Project Karma. So, let's see. Okay, does it work? Yeah. Okay, so you can see here there will be two main parts here, and we will talk about the supporting measure first. So the SIRPa targeting RNP, it will act through the Cas9 clipping and then achieve the SIRPa knockout. So some of you guys may wonder like what RNP is. It's actually the ribonucleoprotein. So we just combine the Cas9 protein and then a guide RNA that targets the SIRPa domain, and then we assemble them, and then we will electroporate it into the macrophage. So it will knock out the SIRPa through the Cas9 clipping. Yeah. So by doing this, we can enhance the phagocytosis. And for the mechanism, we will explain later. And for the main part, the self-polarizing CAR, we will use a siRNA to carry it. So the self-replicating RNA is actually just a vector for the CAR to be transported into the cell. So we will also talk about why we use this RNA instead of other traditional vector like viral vector, or just like electroporating a whole plasmid into the cells. So here you can see the function in circle. So the one we'll talk about the GPC first. So the first function of our CAR is to target the GPC3, and we have searched through online literature and found a SCFB called the GC33, which can target the GPC with a like 80% efficacy, and this will help to target the tumor better, and then also penetrate the tumor microenvironment like more efficiently, and it will also enhance the phagocytosis as it helps the binding of the cancer cell. And for the second function, it would be the expression of interferon gamma. So originally, like macrophage, there will be M0 state. And to differentiate into M1 state, the good one, and the M2 state, the bad one, we'll have to use some cytokine to do so. And interferon gamma is exactly that cytokine. So in our CAR construct, which we won't be disclosing today, there will be interferon gamma sequence attached after the CAR sequence with a P2A clipper. And when the siRNA is transported into the cell, it will be translated using the cell's own machinery, like aka the ribosome. Yeah. So they will express the interferon gamma by the cell itself, and then it can differentiate the macrophage into M1 state. And lastly, there will be also be a small molecule control kill switch. So it's more like an off switch. Whenever there is a small molecule like the one we opt for, concentration, it will stop the transcription of the sRNA, and that will lead to controllable CAR expression. And yeah, we will explain it later as well. Yeah. So for the CAR we use, it will be the third generation CAR. So as you can tell here, the first generation is just like some intracellular stimulatory domain. But for the second generation and the third generation, there will be co-stimulatory domain and also like cytokine inducer. So the one we use is exactly the third generation, in which there will be a cytokine inducer to help with the macrophage function. Yeah. So we are back to the SIRPa part. So you can see that normally the cancer cell, they will evade the phagocytosis through expressing CD47, in which a quite common expressed antigen on their cell surface. So tumor cell will just express their CD47, and then on macrophage, they will have a receptor called the SIRPa. So when they bind together, they will have a don't eat me signal that prevents the macrophage from phagocytosing the tumor cells. So what we do now is just we will knock out the SIRPa so that there won't be the SIRPa antibody expressed on the macrophage, and therefore there won't be the CD47 and SIRPa interaction, and we would prevent the hoving from happening. Yeah. Okay, so by knocking out them, we can prevent the evasion of the tumor cells. So the reason why we do that as a supportive measure is because unlike CAR-T, who target the tumor cells purely through the antibody, the phagocytosis of the macrophage, the CAR-M, is mainly depends on phagocytosis. So to have a good CAR-M, we have to target well, and then we have to phagocytosis well. So we already have a good targeter, but we cannot ensure if it can phagocytosis normally. So this part is just to ensure that it can do it, carry out its function, carry out its duty normally. Yeah. Okay, so I will talk about the sRNA right now. So sorry we didn't show the design of the sRNA here. So it's actually just a RNA construct that would be expressed in the cell, and then there will also be a component in the sRNA per se, which replicate the sRNA. So you can imagine when as sRNA enters the cell, it will first be transcribed, and then also the replicates encoded in the sRNA will also be transcribed. The replicates produced will in turn back to replicating the sRNA per se. So you can see there will be many sRNA amplified in a short time. Yeah. So the reason we choose it over the traditional vector like virus or like the whole plasmids is because there will be no genomic integration. So it should be a safer option where we are going for immunotherapy, and also the manufacturer is quite simplified because it's not a whole plasmid; it's just a short sequence. And then there will also be reduced efficacious human dose. So by this, we mean that we don't need to use too much dosage, like when we are using viral vector, because it will amplify itself inside the cell. So we will just need a small initial dose, and then we will just amplify itself repeatedly, and then there will be a efficacious human dose. And also there will be no viral shell. So unlike using lentivirus or adenovirus to achieve the transfer of the CAR sequence, we use this sRNA, which is without the viral shell, so it can reduce the anti-vector immunity, and there will be less immunogenic reaction as well. Yeah, so this is our project design flow. So first we prepare the cell line. So for the model organism we use, the model cell line we use the THP1 for the macrophage, and then HKU-Hong as the cancer cell that we use for coculturing. And okay, so the second step we will do the SIRPa knockout using the RNP, and we will also construct the sRNA vector. Yeah, and the fourth step we will try to express the CAR in the cell through electroporating the sRNA into the the THP1, the macrophage, and in the end we will co-culture the THP1 and the HepT2 together and do immunoassay to verify the efficiency of it. Yeah. So here's our project novelty. So basically, there is no one who have done the CAR and the SIRPa knockout in macrophage before, and even CAR M per se is a very novel concept. Like for CAR T, there are already clinical trials going on, but for CAR M, they are like very none, and it should be a quite new field to explore. And also for the self-replicating RNA, there are no prior application in immunotherapy before. So for those who know, you might know that the sRNA is being used in vaccines. They do those intramuscular electroporation to get the RNA into the cell and then do a vaccination. But for the immunotherapy, the using of it is never explored before. So we would think that it would be quite a novel approach to tackle the tumor, like solid tumor, like HCC. Yeah. Okay. So here's why we want to do it right now. So first is because the mortality rate of the tumor patient can be reduced, and the reason why we do this is because we can leverage on advanced genome editing tools like CRISPR-Cas9, just like the RNP we just explained to you guys. And also, there will be a novelty approach to overcome the limitation of the CAR T therapy. So this reason will be like why we are going to do this project. Okay, then I will now hand the part to Cyrus.
Alright, so now we'll talk about data collection and analysis. So firstly, we will do sequencing data collection and pre-processing. And we'll get RNA and qPCR sequences for CAR macrophage, coculture with cancer stem cells from publicly available databases. And then we'll perform standard pre-processing steps, such as quality control checks with FastQC, and then align the reads to the reference genome, and normalize the data with delta-delta CT method. And lastly, we can filter out the locally expressed genes. After that, we will move on to differential gene expression and multi-omics integration. So this to identify the DE genes associated with macrophage activation and tumor cell killing. And then by integrating the gene expression data with proteomics and metabiomics data, this allows us to understand the overall molecular mechanism. So we can firstly perform differential gene expression using analysis tools such as DESET2 or H2 and HR and Lima. And then we can use integration tools such as Omic Integrator and Diablo to integrate the gene expression data. Lastly, we can perform principal component analysis and hierarchical clustering analysis to identify the sample clusters, and then we can also do gene site enrichment analysis to identify the key pathways. After that, we can review the potential signaling pathways and biological processes involved in the CAR-M activated immune response. So we can construct a gene co-expression network using cytoscape, and then identify key modules and hub genes in the network. After that, we can perform pathway and gene set enrichment analysis, and lastly, perform network topology analysis. Now I will pass the mic to Duncan.
Hi, I'm Duncan. So apart from the experiments and methodology, let's talk about the education and community outreach. So firstly, we have joined this synthetic biology workshop in Wuxi's campus of Shanghai International School, so that we can promote our project and to share this among the secondary students. And also, we will create some workshop for bioinformatics to create some first-hand experience to secondary students, which is very unique for secondary schools. And also, we'll join the Joint Schools in Science Exhibition, JSSE, to promote our project as university students. And also, we collaborate with overseas iGEM team to make a children's storybook. And also, we have our Instagram account, allowing us to spread scientific knowledge to engage with public. To empower the next generation scientists, we have card game, so that we can balance between scientific knowledge and amusements. And a big thank to the CUHK iGEM team giving us a chance to share our project here. And last but not least, we have the interview with stakeholders just for the integrated human practice, so that we can explore more about our project and its influence on a society's perspective. So to conclude this presentation, we would like to have a big thanks to all your patience and attentive listening. Your presence here signifies your support, not only to us, but also to other iGEM team. And we would like to extend our heartfelt gratitude to the CUHK iGEM team for this symposium, allowing us to share our passion and enthusiasm for synthetic biology here. Thank you.
Please feel free to ask us any questions. I know the presenter might be in a rush, and we might not explain everything very clearly. So yeah.
From what I know, HEC is like it has a hypoxic environment. Like that's what drives the malignancy of the liver cancer. How does your solution kind of address that? Because the hypoxic environment really induces like immunosuppressive molecules or an immunosuppressive environment. So how will you be able to deliver it directly to that environment? It doesn't make sense because hypoxia leads to a very immunosuppressive environment. So how does the macrophage is able to enter it?
So what you have just mentioned there is that in the tumor microenvironment there will be a hypoxic environment that will like very few oxygen, and it will to like harder for the CAR T cell to express their to carry out their function. But for CAR cell itself will have a the one we designed will have the self-progressing CAR, which allows us to transform the macrophage from M0 state to M1 state. And at that stage, the macrophage will be more activated in targeting the tumor cells. So the effects of the hypoxia on the immunosuppression will be suppressed a bit. And we didn't do too much research on the hypoxic environment, but instead the immunosuppression from those tumor-associated macrophage, the M2 macrophage. Yeah. But theoretically, for the enhanced CAR that helps to strengthen the phagocytosis, it can overcome the hypoxic environment.
Very nice presentation and also nice design as well. I'm just wondering, your system using the SIRPa, how efficient is this system to transfer all the components into one cell?
Okay. So sorry, we didn't prepare the slides for the sRNA construct, but still I will just say it from here. Yeah. Okay, so for the sRNA, let's switch to the PowerPoint. Okay, we didn't insert a figure here, but for the sRNA, first there will be a sequence coding for the replicase that helps with the in-cell amplification, and then there will be the CAR construct. So the reason why we didn't disclose our car construct here is partly because it's novelty and also it's being corrected like repeatedly. So there will be a SCMV targeting the GPC3 and then some CD40 and CD28 domain that do the co-stimulation. And then by a P2A cleaving protein, the sequence that once it was transcribed, we will cut off the cell. And then after the P2A region is the interferon gamma. So through synthesizing a plasmid that contains all the components in one, we will then do the in vitro transcription. Yeah. So the promoter will be T7 promoter, and we use the MSHM machine T7 IVT kit to produce that. So as a product, the RNA will contain those all the components we need, and then we will just electroporate them into the macrophage. So for our project, it's the THP1, and we might also do iPSC derived macrophage, depending on the time we have. So thank you guys.